Now we're going to start with the oxidation of pyruvate, but before I get into what it is, I want to review what pyruvate is in the first place. So if you remember yesterday, I was talking about glycolysis and we said that we start with glucose and we end up with pyruvate. Now, today we're going to talk about what's going to happen to pyruvate, okay? So the oxidation of pyruvate, there's this enzyme that is called pyruvate dehydrogenase. It's kind of complex. It's made up of three parts of this enzyme. So I don't want you to know all the details. I just want you to know what I'm going to say right now. Okay, so we start with pyruvate, and then we take carbon dioxide for pyruvate, and this forms. It's called hydroxyethyl TPP. This is where in the first enzyme of this enzyme complex. Okay, decarboxylation, and then what happens is that this forms. Okay. Now, in enzyme number 2, which is the second part of this complex, what's basically the role of this enzyme is to form something called acetyl-CoA. There's a series of steps right here, but she has never asked about them. But it's good for you to know that in this case, we are reducing this lipolysine into this, okay? And then acetyl is added to CoA, and this forms acetyl-CoA. Now, moving on to the third enzyme of this enzyme complex. So basically, the electrons that are found in this complex are then sent to FAD, which becomes FADH2, and then FADH2 relays these enzymes to NAD+, which becomes NADH. That's exactly how this enzyme works. But what is important is that I want you to know that when we start with pyruvate in this enzyme that's called pyruvate dehydrogenase, the products are going to be acetyl-CoA, this is number 1, and NADH. Okay, this is very important. You need to understand and know that we start with pyruvate and we end up with acetyl-CoA and NADH. Now we're going to talk about the regulation of PDH, which is very important. So basically, as I have said before, we're starting with pyruvate, and through the enzyme, which is PDH, we're going to have acetyl-CoA and NADH, right? So this is the old trick in the book. Everybody knows this. If anybody asks you what inhibits this enzyme, you're going to have to say pyruvate becomes acetyl-CoA, and we have increased levels of acetyl-CoA and increased levels of NADH, then most probably they're going to go back and inhibit what? PDH. Check. Now, this slide is kind of a bit complex, but I'm going to try to make it as easy as possible. So imagine that here we have PDH, which is this enzyme, okay? Don't forget, we're talking about this enzyme. PDH is in the middle, right? Now, what is PDH phosphatase and PDH kinase? A kinase is an enzyme that adds PO43- to another enzyme. A phosphatase is one that removes PO43- from an enzyme. So basically what I'm saying is that this enzyme's activity is controlled by either this kinase or this phosphatase. So when this kinase adds a phosphate to PDH, PDH is going to be inhibited. Okay? When phosphatase removes PO43- from PDH, PDH is going to be what? Activated. Okay? This is till now. If you understand this, then you've understood 50% of what I'm going to say. So now, what controls the phosphorylation or dephosphorylation of this enzyme? These enzymes are also controlled by outside factors, okay? So we'll start with acetyl-CoA, NADH, and ATP. These three. So basically, as I have said before, right here, acetyl-CoA and NADH, if their levels are increased, then this will what? Inhibit PDH. So, if these three are increased, they activate PDH kinase. If PDH kinase is activated, it goes and it adds PO43- to PDH. As I said before, if PDH is phosphorylated, this means that it is inactive. Check, makes sense. So I will reiterate, increase in these activates PDH kinase. PDH kinase is activated, it goes and it phosphorylates, or it adds a phosphate to PDH. When PDH is phosphorylated, it is inhibited. Okay? Now, what inhibits PDH kinase? If I say this enzyme is being inhibited, this means PDH is going to be activated, okay? Because it can't come and add PO43 minus to it, so this one is going to stay activated. So what are the factors that inhibit PDH kinase? We have calcium, pyruvate, and dichloroacetate. Now, a trick for you to remember why calcium actually inhibits PDH kinase is that if you remember in muscle contraction, calcium plays a very important role in the process of contraction. So basically, let's say you are active, then this definitely will increase your calcium levels in the blood. And these calcium levels means that the body wants more energy. The body wants us to produce more energy. So if you want more energy, then that means you want this cycle to happen, because when acetyl-CoA is formed, it then goes and forms ATP, right? So when the body wants more ATP by increasing calcium levels, because you are working out, for example, then this means this is favored. I want this to work. So I want this enzyme to be activated. So calcium comes, it inhibits PDH kinase, and when this one is inhibited, it cannot inhibit this one, so this one is activated. And it's the same case for both of these, because if pyruvate is increased, it means that I have a lot of substrate that needs to be what formed into the product. So then that is also a stimulation for it's an inhibition for PDH kinase, which is a stimulation for PDH. So what is PDH phosphatase? PDH phosphatase, as I said before, it removes a PO43 minus from this PDH, which means it activates PDH. So what activates this enzyme to activate PDH? Magnesium, calcium, and insulin. Magnesium is actually a coenzyme or a cofactor that is needed by enzymes. So when we see that there's high levels of magnesium in the cell, this means that the cell or cellular respiration is favorable, because many enzymes of cellular respiration require magnesium to work. So this makes sense that magnesium would actually activate this guy, which will also activate this guy. Okay? I had already explained how calcium works. Here it's inhibiting PDH kinase because PDH kinase inhibits PDH. Here it's activating PDH phosphatase because it wants to activate PDH. Now, insulin, as I've said in the last lecture, I'll go back to it. If you remember yesterday here, I talked about how insulin binds to its receptor, and this leads to the formation or the fusion of the vesicle with the plasma membrane. So basically, insulin here is increasing these GLUT receptors, and glucose is coming in, so it's promoting glycolysis, right? So if I come back here, we remember how insulin works, then this is most likely to activate PDH, right? Because when insulin is activated, this means we have an increased energy demand for ATP, which means this reaction is favorable, which is this reaction. This reaction is favorable when insulin is increased in the blood. Okay, now why does NADH, NAD plus inhibit PDH? Hint, think in anaerobic conditions. In anaerobic conditions, as we remember, glycolysis does not need oxygen to work, right? But after glycolysis, we have pyruvate. And if pyruvate wants to enter the citric acid cycle and into the mitochondria to have ETS to generate ATP, here we need oxygen. So it's telling you that NADH inhibits PDH in anaerobic. So we don't have this process. So we're entirely depending on glycolysis. Since the cell is entirely depending on glycolysis, then that means it has an increased demand for what? NAD+, right? I'll go back and I'll remind you. So basically, if you remember, in the sixth step of glycolysis, we need 2 NAD+, and these NAD+ are going to be reduced to NADH, okay? So when it's telling you that you have an increased NADH over NAD+, and the cell is dependent on glycolysis, then of course this is going to inhibit PDH, because NADH is not needed in glycolysis. What's needed is NAD+. NAD plus is needed for glycolysis, and I'll reiterate, if it's an anaerobic condition, then of course it's only going to have glycolysis, and in glycolysis we only want NAD plus, okay? So if you activate PDH, you're going to take this NAD plus and make it NADH. But since you want NAD plus, you can't have this, so this should be deactivated. So this should be inhibited, okay? Now, what about the role of AMP in this regulation? So this is a trick for you to remember. Anytime you see the word AMP or adenosine monophosphate, this is going to tell you that the cell is having low energy levels. If you see ATP, then this means high energy levels, okay? So AMP, as I said, low energy levels. So what is it going to activate? Of course, it's going to activate glycolysis, right? Because glycolysis is going to give us 2 ATP. Okay, moving on. Okay, so we will always be asking this question. What is the fate of pyruvate? We have 3 fates for pyruvate. These 2 depend on the presence of oxygen, okay? If there is no oxygen, just as I have said yesterday, pyruvate is converted to lactate via this enzyme, lactate dehydrogenase. If there is oxygen, then there's 2 pathways pyruvate can go into. It's either gluconeogenesis or the citric acid cycle. And that is entirely dependent on the concentration of acetyl-CoA. The more acetyl-CoA you have, this indicates the more ATP the cell has. This indicates that the cell does not want any more ATP or energy. So, of course, PDH is going to be inhibited. Why? Because PDH converts pyruvate to acetyl-CoA, which is then energy, or ATP, you could see right here. So, increased levels of acetyl-CoA is gonna inhibit pyruvate, and at the same time, it's gonna stimulate pyruvate carboxylase, which converts pyruvate back into glucose via gluconeogenesis. And this extra glucose that we have can go to the liver to be stored as glycogen or to form fatty acid, to become fatty acids. Okay. Now, how can the electrons reach the mitochondria? As I said before, in the glycolysis and in the citric acid cycle, we have NADH and FADH2 that are holding these electrons that are formed from these reactions. But how do these electrons actually reach the mitochondria? Well, the answer is this shuttle or the other shuttle that I'm going to talk about in a second. So, glycolysis, as I've said before, generates NADH. This is the one that's holding the electron. NADH then sends this electron to DHAP. Since DHAP cannot actually enter or cross the mitochondrial membrane, it becomes G3P. G3P then holding the electron from NADH delivers this electron to FAD. FAD is then reduced to FADH2 and the electron is delivered to the mitochondria, and then in the electron transport system we have the oxidative phosphorylation and then ATP is produced. This is the second shuttle called the malate aspartate shuttle. So as I've said before, we always start with NADH. NADH is the one that's holding the electrons that are going to be delivered into the mitochondria. So basically, we're going to start with NADH giving oxaloacetate a hydrogen. Since oxaloacetate cannot come into the cell, it becomes malate, into the mitochondria, I mean. It becomes malate. Malate then can cross into the mitochondria. Malate then, since it took the H from NADH, it returns it back to NAD+. Then malate comes back to oxaloacetate, and NAD+, which took the H, becomes NADH. Okay? So we delivered the electron into the mitochondria. Now oxaloacetate has to go back out, right? So, as I said, oxaloacetate cannot cross. It needs to be converted to something else so that it can leave, right? So what happens is that glutamate donates this amine group to oxaloacetate, and oxaloacetate becomes aspartate. Glutamate, when it donated this, it became alpha-ketoglutarate. Okay? I'll repeat. Glutamate donates NH3 plus to oxaloacetate. Oxaloacetate now becomes aspartate, which can easily diffuse out of the mitochondria. As for glutamate, since it donated NH3 plus, it becomes alpha ketoglutarate, which leaves the mitochondria right here because it wants its amine group back, okay? This is very important. Energy is neither created nor destroyed in chemistry. It's just transferred, okay? So glutamate does not give this amine group to oxaloacetate forever. It is turned to alpha ketoglutarate, which goes back outside and waits for aspartate to leave so that it can return the amine group back to alpha ketoglutarate. Okay? Alpha ketoglutarate moves out of the cell, waits for aspartate to leave the mitochondria and return this amine group to alpha ketoglutarate. When this amine group is returned to alpha ketoglutarate, it goes back to becoming glutamate, and of course, when you remove this from aspartate, it goes back to oxaloacetate. Okay, so this is the malate aspartate shuttle. These two are important. These are very important. Okay, guys? Guys, this is very important because she actually asked about this last year. So basically, what is the direct product of glycolysis? As we said before, 2-NADH to ATP. This is the pyruvate oxidation. We talked about this before, 2-NADH. And I don't know if you took this yet. The citric acid cycle gives us FADH to NADH to ATP or GTP. So if you want to calculate how many ATP you're going to have at the end of cellular respiration, you're going to have to calculate how many ATP NADH and FADH can yield, okay? So, if 2 NADH can give you 5 ATP, then that means each NADH can give you 2.5 ATP, okay? And if FADH2 can give you 3, then that means each one can give you 1.5 ATP, okay? So, if she asks you in the exam how many ATPs are produced due to pyruvate oxidation only, so what you're gonna think is that, okay, pyruvate oxidation means that I'm gonna have 2 NADH produced. If I have 2 NADH produced, how many ATPs can one NADH form? So it's 2.5, but it's 2, so it has to be 5. So the answer to this is 5 ATP, okay? And if you add all of these ATPs that are formed, you're gonna have 30 or 32. Now you're gonna tell me, why is it not one number, and why is this 3 or 5? So this actually entirely depends on the efficiency of the cell to shuttle or send NADH's electrons into the mitochondria, okay? It just depends on the efficiency and if the cell is working hard or not, okay? I hope this makes sense. So this is easy. It's telling you what's the significance of the malate aspartate shuttle. We need to deliver the H plus to the electron transport system. We should let go of this H so that NAD plus can go back to glycolysis. As I told you before, NAD plus is needed for glycolysis, and these two shuttles are found in many tissues. So basically what this page is talking to you about, if the cells undergo hypoxia, what's going to happen. It's very understandable if you read them. I wrote them in a very nice way, and please don't forget to read the notes that I write in the description because they explain everything that I'm talking about, okay? Now this is very important because a question came from this last year in our exam. So what is the Pasteur and Warburg effect? The Pasteur effect, if you introduce oxygen to an anaerobic cell, then this cell is going to switch from anaerobic respiration to aerobic respiration immediately, okay? And then this guy observed that glucose consumption is increased in anaerobic conditions. Now, what does that have to do with tumors? So basically, if you remember what a tumor is, the abnormal growth of cells, it's growing to a point that the capillaries cannot keep up with the supply to this tumor. So what's going to happen is that the tumor is going to live in hypoxic conditions, right? And if I recall, and if I ask you now, what type of respiration does a cell depend on if it has low amounts of oxygen? You're going to have to answer glycolysis, right? Because glycolysis does not need oxygen. And if tumor has low amounts of oxygen, then that means it's going to depend entirely on glycolysis. So what does that mean? It means that it's going to have increased glucose consumption, okay? It's going to take so much glucose because it needs so much ATP, and glycolysis only one glucose can only give you 2 ATP through one glycolytic pathway. So imagine the amount of glucose this tumor is going to need to have enough energy for it to function. Okay? Now these are notes about what I have just said. Read these two because they are nice to know. This is gonna tell you about the types of mutation that can happen for the tumor to form. Read them too, please. And for the diagnosis of certain types of cancers, we're gonna need to have positron emission tomography. Please read those too. These are the clinical aspects of this lecture. And thank you so much for listening, and I hope this was very helpful for you. But for now, I put these questions for you to practice on. They're not easy. I'm telling you, I'm giving you heads up. And this is a question that we had last year from our exam. I hope you answer it right and you know why the answer is right and you've understood everything. That's it.